All right, diving back into it for part 3, we're going to pick up where we left off, thinking about how we can see changes over time in a population. Okay, so this slide is about the heterozygote advantage, having an advantage of being heterozygous for a certain trait. Okay, so again, just kind of a reminder. Only expressed alleles are subject to natural selection, meaning when a gene is expressed, it makes a phenotypic difference. It makes a difference in the phenotype, and that phenotype is what is going to bring on the advantage or the disadvantage. Okay? But when you're a heterozygous for a particular trait, you're actually a carrier of an unexpressed recessive allele. Does that make sense? Right? So remember, to express the dominant trait, you could be homozygous dominant and have 2 dominant alleles, but many times, if it's a dominant complete pattern, if you're heterozygous, you have 1 dominant allele and 1 recessive allele, and that recessive allele is hidden. That increases the genetic diversity in the gene pool without changing what the population looks like. If the whole population shows the dominant trait, you don't know what % of that population is homozygous dominant versus heterozygous. And those recessive alleles, when they get expressed, because an offspring gets 2 recessive alleles, 1 from mom, 1 from dad, sometimes that is a way to show greater fitness. Maybe the recessive trait is actually more beneficial than the dominant. Okay? So when there's a lot of heterozygous individuals, you'll have these times, 25% of the time, if 2 heterozygous individuals mate, 25% of the time they'll have an offspring with the recessive trait, just like we saw in Mendel's peas. And so when the environment changes and now that recessive trait is beneficial, we're going to see a change very quickly in the gene pool, and we're going to see that recessive phenotype become more common. However, sometimes the recessive trait is not beneficial, but yet it's still beneficial to keep some of it around, and so we call this balanced polymorphism. So that means that there's always going to be this ratio of dominant and recessive traits. So we're going to be looking at sickle cell for a minute, and we've mentioned this in class a few times, and this is one of the options of the video for you to watch for lab. There's one about malaria and being heterozygous for sickle cell disease. So you might check that out, and if you already have, then this might make a bit more sense to you. So folks that have sickle cell disease, and I will say there's a few different types of sickle cell, and so this tells you that this is talking about HB-HSBS, which is just a specific variation of sickle cell. Folks that have this type of sickle cell, they're not going to live to the life expectancy of an average human, in the 70s. They tend to die early because of complications with the disease. So and it is a recessive disease. Heterozygous individuals have 1 normal and 1 mutated, right? These individuals show an intermediate phenotype, meaning their blood cells will sickle, but only at low oxygen concentrations, meaning when they're working out really hard, maybe when they're up at a certain altitude, it's only a temporary sickling of cells, and then they go back to normal when they're in an environment with a sufficient amount of oxygen. Versus if you have 2 recessive alleles and all the time some of your blood cells are sickled. So we see that you would think that the trait for sickle cell, since you die early, right? That gene, the allele, the recessive allele, would just eventually kind of work its way out because of lack of reproduction, or lack of people getting to the age of reproduction, or people choosing not to reproduce because they knew they would be passing on this disease. However, In regions of Africa where malaria is present, we find that we have a higher frequency of sickle cell because of the advantage of being heterozygous. Okay? So malaria is caused by a parasite, and it's passed by mosquitoes, but it's when a parasite invades your body and attacks your liver, and it destroys normal red blood cells. Okay? But it seems, with more research, that the parasite can't attack red blood cells of heterozygous individuals. It's that sickling or some other change in the red blood cells, they just don't seem to get malaria. They're immune to it. They're resistant to it, which is kind of interesting. So even though being homozygous recessive, having sickle cell disease is selected against, because obviously you don't live a full life and you have pain and issues all the way through your life, but it's maintained in the gene pool because in parts of Africa and other parts of the world that are dealing with malaria, being heterozygous, meaning normal blood-wise, most of the time you're not going to have many attacks of sickling of your blood cells, but it's favored because you are immune or resistant to malaria, which is also a fatal disease in Africa. So that's kind of a cool, it's an interesting thing to think about. Now I know it shows the genotypes here that normal, just look at the A's, okay, the A's and the S's. So big A is normal, and then the S is for sickle cell, okay? And so if you're heterozygous, you have kind of the best of both worlds. You don't have full-blown sickle cell, and you can't get malaria. The problem is you also have a chance of passing it on to your children, who, if you think about that Punnett square, if 2 heterozygous individuals reproduce, 1 out of the 4 will be completely normal, 2 out of the 4 will be carriers and have the best of both worlds, but also 1 out of 4 will have sickle cell, or at least that's the probability. So, I know I've said this over and over again, but it's worth repeating. So microevolution is the idea of little changes. Micro means little. And here's the thing you have to understand. Individuals do not evolve. Individuals do not adapt. We don't say it that way. Okay? The individual in the population has certain traits, certain genotypes and phenotypes, and some of that gets passed on to offspring. So when we look at evolution occurring, we're looking at how the genotypes and the phenotypes change in the population. Okay? So again, a definition of a population, in case you don't remember it, that's one species. Okay, we're not going to look at all the living things in a forest. It's just one species that are in the same area at the same time, and they can reproduce with one another. So this idea of microevolution is when we see these small but measurable changes in the population over the course of generations. And it may be quite a bit of generations, but sometimes it's not. Again, going back to the start of this, Darwin noticed this, and it's still the foundation of natural selection. Members of any population have variety. Okay? And I know when we talked about genetics and Mendel, we kind of simplified things and said we have a dominant allele and a recessive allele. But in the real world of genetics, there's actually many different forms of the allele. There could be multiple dominant alleles, multiple recessive alleles. And so that shuffling at a population level can result in a range of phenotypes. Okay, and we can also remember that, hey, sometimes a phenotype is not just one gene working. It's not just that you're yellow or green, right? That there's these polygenic traits that work into it. Okay, so I've said this word gene pool before, and a gene pool is just if instead of looking at the individuals, you look at all of their genes. And usually when we think about a gene pool, we're just focused on one gene or one trait. Okay? kind of like when we look at a pedigree chart and we just follow that one trait through the family tree. The gene pool is just talking about all the variety of alleles that are possible to come out as individuals as they all get shuffled around. Okay, and what we'll look at is we look at the frequency of certain alleles and how they change over time. So if we have 100 individuals, that means we have 200 alleles. Does that make sense? Because each individual has 2 alleles for every trait. One from mom, one from dad. So out of 100 individuals, we have 200 alleles, and we want to know, well, how many have this dominant allele? We'll say dominant allele number 1, or dominant allele number 2, or recessive allele number 1, recessive allele number 2. And if we can take a survey of what that population, that gene pool looks like now, and compare it to what it looks like after 1 generation or 2 generations or 10 generations, if we see a change in that frequency, that's enough to say that microevolution has occurred. Okay, so we talked about industrial melanism in one of the other videos, how because of the Industrial Revolution, much more coal was being burned, soot in the air, and so trees and other surfaces became darkened. So we're going to look at the peppered moth as kind of a classic example here. And so we're going to use the capital letter D for dark, dominant, of course, because it's capital, and lowercase d for light coloration. And so if we looked at a population of peppered moths, we could identify how many big D's and little d's there are in the population. And again, we're going to keep this simple, that we're only thinking one or the other. Something that we assume in some of these gene pool simulations is that there is random mating, meaning there's not mating for life or monogamy going on, that it's pretty random. We tried to simulate this a bit in our paper airplane activity as well, where you just were supposed to replicate the plane of whoever was closest to you. Okay? And so what we can see is that within the gene pool, if there's random mating, there's really no change of allele frequency. We're just mixing them up. Think about having a deck of cards, and you have 52 cards, half are white, half are red, half are black. If we shuffle that deck of cards, the order of the red and black will change, but they're still all there. Does that make sense? So the allele frequency is going to remain the same even through sexual reproduction. That alone doesn't bring about a change of allele frequencies, okay? That's just shuffling around the alleles that we have. Also, even though we have a trait that is dominant, doesn't necessarily mean that over time it will become more common, okay? Don't worry about the little math equation there. That's not really important for us. So I've got a little picture of my peppered moths here. So we have a dark moth on a lichened covered tree, and you can see he stands out. If I was a bird, it'd be easy to see him. Did you even see the light colored moth? He's down here, nice and camouflaged, right? However, through industrial melanism, the tree's darkened. You can see that the lichens are all dead, and now this is just the dark bark. And that light colored moth sticks out like a sore thumb. And the black one, I can still see him, but he is definitely better camouflaged than the white. And so the thing is, this happened kind of quickly over time, the change of the tree coloration, and so we saw the change of the peppered moths happen over time. I don't really want to get into Hardy-Weinberg, but there's some guys that came up with an actual calculation for how to see the frequency of dominant and recessive alleles changing over time. So we're going to see this, but I really want you to just know this more conceptually than mathematically. And so here's the thing. If we add up, this is kind of like the FOIL method, if you remember that from algebra, but essentially if you add up all the frequencies, you're going to have some that are homozygous dominant, right? P squared means 2 dominants. You're going to have some that are heterozygous, and you're going to have some that are homozygous recessive. And when you add them all up, you're going to have a complete gene pool that adds up to 1. And so when we look at the peppered moths, we find that the dark coloration is dominant, We have to do some math and some research to figure that out. Oh, I'm running out of time, so I'm going to pause here and we're going to get started on this slide in just a second.